Thank you very much. I've been really following the rise of the private space industry with growing excitement over the last at least 10 years or so. So I really appreciate the opportunity to be here and to be on a panel with such esteemed panelists here today. So quick show of hand from the audience. Who here has seen the Sci-Fi Channel series, The Expanse? Awesome. 2. So that proves my hypothesis that freelance journalists watch more TV than rocket scientists. So what really interests me about The Expanse, it's a series set some 200 years in the future. It has its share of political intrigue. It's got some good gun battles. But as a business writer, what intrigues me is that it's a very detailed picture of an off-Earth economy. So in The Expanse, we see harvesting ice from the rings of Saturn. We see companies mining asteroids for precious minerals. We see salvage operations, vehicle construction and repair happening in deep space. Routine transport of passengers and cargo between far-flung outposts of humanity, and the challenges of growing food and sustaining human life long-term in a microgravity or zero-gravity environment. So as I mentioned, The Expanse is set 200 years in the future, but from everything we've heard in the last few days, particularly the talk yesterday from Planetary Resources and Peter Diamandis' overview about exponential change in technology, I wonder if we're really that far away from the world of The Expanse. Indeed, we already have a thriving space economy. We've probably heard more than once this week, the global space industry has grown to over $330 billion annually. Most of that, about 3/4 of that, coming from commercial space activities. More venture capital was invested in the space industry, the US space industry in 2015 than all the previous 15 years combined. NASA has played a key role in creating this economy, and it's poised to play an even greater role in accelerating it. So you've heard a lot about the incredible research, the range of research and commercial activity that's already taking place aboard the International Space Station. Designated National Lab in 2005, the ISS will now serve out the rest of its useful life, not only as a platform for innovative research, but an incubator for businesses that can benefit from microgravity and other unique aspects of low Earth orbit. So really the hub of an off-Earth economy comprising life sciences, manufacturing, material science, remote sensing, satellite launch services, and more. The on-ramp to the ISS is CASIS, so the nonprofit founded in 2011, and tasked with really managing and facilitating commercial research aboard the ISS, which can include coordinating launch services for users, providing technical and logistical support, identifying partnerships, funding opportunities and markets for LEO-based products and services. So our 3 panelists today represent 3 key initial focus areas for low Earth orbit commercialization: tissue engineering, protein crystal growth for drug development, and on-orbit manufacturing. Apart from their benefits for humanity, these are huge economic opportunities. The global market for tissue engineering and regeneration is forecast to reach nearly $60 billion by 2019. Obviously, global pharma sales are huge, over $1 trillion annually, and worldwide revenue from 3D printing is forecast to reach $20 billion or more by the year 2020. So in the next hour or so, Dan Tagle, Joseph Ng, and Andrew Rush will offer us an overview of their work in microgravity, and we'll have a conversation about their experiences working with CASIS to advance their research aboard the ISS and the capabilities of the current ISS crew and facilities. We'll discuss the business case for working in space and hopefully get to brainstorm a bit about what the future may hold. We'll leave time at the end of our discussion for your questions. So let's do some introductions. Dan Tagle, at my left here, is Associate Director for Special Initiatives at the National Center for Advancing Translational Sciences, NCATS. Recently, he served as Acting Director of NCATS Office of Grant Management and Scientific Review, currently serves as Executive Secretary to the NCATS Advisory Council and Cures Acceleration Network Review Board. Good title, Dan. So prior to joining NCATS, Dr. Tagle was Program Director for Neurogenetics at the National Institute of Neurological Disorders and Stroke, where he helped develop programs to examine genomics-based approaches in studying inherited brain disorders. He's an accomplished researcher in his own right. He holds a PhD in molecular biology and genetics from Wayne State University School of Medicine, was a postdoctoral fellow in human genetics in the lab of Francis S. Collins at University of Michigan, and as an investigator, section head of molecular neurogenetics at the National Human Genome Research Institute, was involved in a collaborative effort identifying molecular markers for genes involved in Huntington's disease and other disorders. Dan's the author of more than 150 scientific publications and has garnered numerous awards and patents. And I'll just introduce everyone as we move through here. Joseph Ng is a professor in the Department of Biological Science at University of Alabama, Huntsville, where he is principal investigator for a research team studying structural biology and director of the school's biotechnology science and engineering program. He's also co-founder and president of iExpress Genes, a private biotechnology company that provides, among other things, protein purification and crystallization services for a range of commercial, institutional, and academic customers. He has nearly 20 years of experience in microgravity research, having conducted numerous protein crystallization experiments aboard both the space shuttle missions and on the International Space Station. He's also worked with the French Space Agency as a postdoctoral fellow. He's been instrumental in advancing the biotech community in Huntsville through his involvement with the UAH Business School, private companies, and government laboratories. And he really brings an international perspective here, having worked, as I said, in Europe with European scientists and recently returned from teaching a crystallization workshop in the Czech Republic, very recently, so if he's tired, we can forgive him. Finally, Andrew Rush is the president and CEO of Made in Space, a Silicon Valley-based company that designs and operates additive manufacturing or 3D printing technology for use in space and other extreme environments. His company has clients who include NASA, as well as the home improvement chain Lowe's, and it's the first to do commercial manufacturing in space with its new additive manufacturing facility recently installed aboard the ISS. Andrew was previously an intellectual property attorney specializing in aerospace and additive manufacturing. Actually joined Made in Space as its general counsel. He's worked at Maston Space Systems, and prior to getting his JD, he earned a BS in physics and worked as a research assistant in a solid-state physics laboratory. So, great panel, and I believe Dan has offered to start us off by giving us a brief presentation on his work.
Thank you, Adam, and certainly thank you to the organizers, especially CASIS, for this opportunity to present this morning. I kind of lead off better, I guess, this morning, because I'm the least experienced. We haven't really done any lower Earth orbit research, but certainly have now been in partnership with CASIS, which I will describe in this presentation, especially on microphysiological systems, or tissues on chips, or sometimes called organs on chips. This is an attempt to take this technology that we have developed for use here on Earth to advance drug development and then hopefully be able to deploy for research at the International Space Station to better advance health here on Earth. It's a fairly complex program, the microphysiological systems, and so what I had done was essentially to be able to show you a video, a short video that would highlight what this program looks like. So if we can have audio on this, please.
Several thousand diseases affect humans. of which only about 500 have any treatment. Getting a drug through the current development pipeline takes too long, is too costly, and the vast majority of new therapies, about 95%, never make it past clinical trials. Drugs that seemed promising in early studies using cell and animal models often don't have the same response in humans. We need an alternate research model that accurately represents the human response. NCATS, the National Center for Advancing Translational Sciences at the National Institutes of Health, is supporting research to make this possible. Through the Tissue Chip for Drug Screening Program, a collaboration among NIH, DARPA, FDA, and researchers nationwide, NCATS is supporting research on bioengineered devices that mimic the function of human organs. The goal is to use these tissue chips to test the safety and effectiveness of potential new drugs, weed out those that don't work, and pursue ones with the most promise. The chips are fabricated from a flexible plastic with ports and channels to allow nutrients and oxygen to pass through them. Unlike cell culture and animal models, the innovative design of tissue chips replicates the complexity of the human body environment. One example is a liver chip, which includes multiple cell types that are organized much in the same way as they are in the human liver. Features such as blood flow can be mimicked to recreate what cells experience in the body. The complexity of these chips generates human-specific data, which enables researchers to better understand how the liver responds to a drug and if it is toxic, critical data before testing in people. By connecting chips that represent various organs into a full human body system, scientists may better understand how the whole body responds to drugs and if they're safe and effective. For example, chips that include additional factors such as hormones could help researchers investigate differences between male and female responses to drugs. The ultimate goal is to use these chips in a variety of ways, including to advance precision medicine, test for environmental toxins, and develop disease-specific models such as for cancer and rare diseases. By developing, demonstrating, and disseminating tissue chip technology, NCATS is improving health through smarter science.
Yeah, so one of the main goals essentially that we have in this program, so the program's been running since 2012, and certainly have made several advancements in terms of developing this technology. So what we are wanting to do now is to use this tissue chip technology instead of using drug exposure as one of the parameters to test. Now we're looking at the effects of microgravity, perhaps extreme radiation exposure, and essentially looking at the genomics, metabolomics, proteomics that would alter human physiology and response. And so in this particular slide, I'm showing you examples of the tissue chips that we have developed so far in this program. These are just a few representatives. So the one in the bottom right, as you can see, is our gut enteroids that's pulsing. So these are actually innervated guts, and they pulse because there's peristalsis in response to kind of like digestion. So obviously in the chips there's no food, but they do pulse. And then as you see the release of lysozymes, digestive enzymes in this gut as well. Our investigators have also been able to grow the microbiome in this gut on chips. On the top right would be a microvasculature. You can probably, I don't know if you can see it from when you're sitting, but there are 1 micron microbeads that are flowing on those, in this microvasculature. So it's a way of perfusing these 3D constructs. Next would be, right in the middle, would be a muscle bundle that is fluorescing, that shows calcium release as a function of contraction. So these muscle bundles actually are tethered to cantilever device that not only measure force contraction, but they also are able to give us very precious information in terms of muscle activity and so on. And then on the top left, I don't know if it's showing, well, maybe not. But the top left actually would have shown, if we can go back, the top left, oops, sorry. Can we go back to, would be cardiomyocytes that would be beating, slow beating because it's from a diseased cardiomyocyte from a long QT patient, and then you treat that with isoproterenol, it will accelerate the beating. So the partnership that we have with NCATS, as you know, NIH is the largest biomedical funding agency in the world. We have a budget of $32 billion each year. And so NCATS, in partnership with CASIS, has formed partnership through a memorandum of understanding with the sole purpose of deploying this technology for use at the International Space Station. So the expected outcome is that we will be able to understand more about diseases and conditions that affects the bone, the cartilage, the muscles, the GI system, the immune system, the microvasculature, and other tissues that are known to be susceptible to prolonged exposure under microgravity environment. The potential impact, of course, is that we will be able, through analysis of the epigenetic changes, some of the molecular signatures that would characterizes changes in the human body when exposed, in the human tissues when exposed to space, will provide us with examples or perhaps novel targets for drug development that can be used here on Earth. So, and in some ways being able to either slow down or mitigate the effects of aging. And so the partnership that we have will be a request for applications to tissue chip developers to partner with CASIS space implementers and to be able to work together and to be able to deploy this technology into the International Space Station. We're expecting the funding opportunity announcement to come out sometime in the fall, probably in September, with the application receipt date being in December and the award date July, and the project to start in August of next year. More information can be obtained at the blog spot that I indicated in the bottom corner of the slide, or I'd be happy to answer any questions later on as well. So with that, I thank you for your attention.
Thank you, Adam, for the previous introduction, and thank you for the organizers, in particular CASIS. I want to talk about our previous flight experiment, and if I could have the first slide. So we are in the structure design-based drug discovery business, and let me just begin by telling you what is entailed in this. If we are to design a drug against a disease, we start out by identifying the disease, and we look at any particular genomic sequences or protein sequences that are associated with this disease. And the idea is to try to design things that would inhibit its action by looking at its structures. And in order to do that, we use a technique called X-ray crystallography, or crystallography in general. And what that is to look at diffraction analysis to determine three-dimensional structures. Now this scale here, this routine here that you've seen is from crystals to structures. And this is important because this structure is the starting point for any type of drug design. And the accuracy of this is really important for any type of specificity for a certain drug. And so for a drug to go out into market, it not only has to demonstrate specificity, it has to demonstrate that it is safe. So when you see a drug commercial on TV, and you see all this nice, beautiful scenery, green, and couples dancing with each other, and all of a sudden at the end, you see these horrible side effects. And these horrible side effects, as alarming as they are due to the specificity of the drug. So the more specific a drug is to a particular target, the less perhaps side effects that it may have. So this is why it is important in the molecular level to look at the structures. But let me point out the important steps here, and that is at this design. You can't be able to forward this next slide. Is it possible to move the next slide? Yes. So this is give you an idea of the cost behind a drug design studies, and this is why medication costs so much. The computation modeling is relatively cheap here, and this relies on the accurate structure model. And this is what we're trying to do, is trying to obtain the most accurate structures that you can at an atomic level. Thereafter, it goes through biochemical studies and assays, and as you can see, it goes through cell cultures, and then thereafter animal studies, and after human. The prices really goes up, as you can see, and this is why it's so costly for any drug design to occur. But the accuracy is, again, as I stated, it depends on the accuracy of deciphering the atomic structure of the targeted protein for an associated disease. So just to give you an idea of how many structures are there out in the public protein database bank, there are over 100,000 deposited structures. That is, that these are three-dimensional coordinates, all of them that potentially can be associated with some kind of life science function or diseases. And these are what drug designers would do. They look at structures that are out there, they look at three-dimensional structures. But these are performed by X-rays, and X-rays reveal carbons, nitrogens, sulfurs, and oxygens. And this is just the chemistry 101 for you, is that all proteins are made out of that, but it does not reveal hydrogens. And hydrogens are revealed by neutron crystallography, and this is why we are growing large crystals. And just to give you an idea in comparison, there are less than 100 structures that's showing neutron structures which revealing hydrogens, which is imperative for drug designs. And out of that, that's a very, very small percentage in terms of overall structures of this. So neutron crystallography is a word you're going to hear in the next minute or so. And 50% of the atoms in proteins are hydrogens. They are invisible to most structures that's been solved. So most of the structure-based design that's looking at these are blind to these hydrogens. And this is why it's valuable to do these neutron structures. The neutron crystallography structures will reveal hydrogen atoms, and this is just showing a display of the electron density, which are the blue cages that you see, the positions of these hydrogens. And these are imperative in knowing the exact details of how to design a structure. So we had sent up an enzyme, a protein called inorganic pyrophosphatase. This is a potential target for antibiotic, and as you may know, the antibiotic resistance is a big problem here. And this is a protein that is responsible for hydrolyzing this reaction from inorganic pyrophosphate to pyrophosphate. So this is the only biochemistry lesson you'll see here, but the important thing is that this enzyme, if we are able to find an inhibitor for it, is a potential antibiotic. So we use the hardware called the gonadocrystallization facility, which is the GCF. It's a passive box which contains these plastic cases called the GCBs. We like to use all these different acronyms here. That stands for the gonadocrystallization box. In short, what they are is we put capillaries inside these trays, and the whole idea is to have protein solutions diffused against a precipitant in such a way that crystals will grow. Well, we're out to grow large crystals. We're not out to grow any crystals, but big. We're thinking big, 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 big volume, big crystals. And the reason is because neutron crystallography requires to have a large crystal. So 1 cubic millimeter is the size that we're required to grow. And so this is very difficult to do it on the ground. And this is what you see. This is an experiment we sent up on with SpaceX 3. We had an experiment going up there for 6 months. This is an example that was brought down immediately in a hotel room that was retrieved. And this is with no magnification. This was with an old iPhone 5. Just looking at it in a hotel room. We're very, very excited that we're seeing these large crystals. These diameters of the capillary were 2 millimeter, and that's considered really, really big because you don't require a microscope. Just to give you an idea of the differences between earth-grown and microgravity grown crystals, to your left are the ground ones that we've grown. These are just parallel experiments, same protein, same setup, same people, same place, same cargo, everything, except one's on the ground, microgravity. As you can see readily that on the right, microgravity shows these really pronounced large crystals growing out the diameter of the capillary. Again, these are 2 millimeter. This is really unprecedented in how big they are. Here's another example, ground on the left and microgravity on the right. These are statistically significant because each sample, we've set 5 redundant samples of the same thing growing. So you can see there's a drastic effect in our case in terms of using the ISS platform to grow these crystals. This is an example of what these crystals look like. Again, they grow out to over 6 cubic millimeter. That's actually very, very big. I know you guys are used to diamonds and things like that. For in the protein world, anything over 1 cubic millimeter is an astounding accomplishment. So just to give you an appreciation of that, this we published already, so it's actually made a lot of posters. And so just to give you an idea of the quality of these, again, I mentioned diffraction. Diffraction is based on intensity over the resolution. So this graph tells you 2 things. The space ones diffract better, and it gives you higher resolution, meaning that you can see the atoms much more clear and highly more resolved from those that are grown in space compared to that of Earth. Here's another one, again, the same thing. These graphs of space and Earth shows higher intensity and higher resolution. What does that translate to? In terms of looking at the clarity of an atom, this is just a location of this. Space, you could see more densities, you could see the assignment of where the atoms may be, where in the ground, where you have less quality crystals, you may not see these. And for neutron diffraction, this is an example of what a neutron grid may look like. I don't have time to go into details, but just to say that growing large volume crystals permit us into utilizing neutron crystallography to locate hydrogens where otherwise it would not be possible. And this is just an example. To your left, you see the orientations of water molecules. These are important for us in terms of knowing what they interact in for drug designs. On the right is just X-ray structures, so you don't get the details of this. And I'll show you an example of what I mean. So in this last slide, I want to show you the mindset of what a structure-based design will look like. So this is a movie, if we could just play it. This shows the atomic structure that we've got from a composite from space-grown crystals and X-ray structures that we've gotten on the ground. What you're seeing at is zooming in into what is called the active site of this particular enzyme. The idea is to design something that will block this active site. And these cages tells you the distribution of the atoms. The more clearer and outlined the cages are, the more accurate they are. So from a pharmaceutical perspective, if you are to design something, you want very, very clear outlook of what you're going into. So it's like building, remodeling a house. You need blueprints, you need details of things you want to build, and this is what we want to do with molecular structures. And we want to dive right in. If we're going to design something, and if we want less side effects, we want more effectiveness, we need to know exactly where the locations are, the atoms, and have a nice specific fit. And so by growing these large crystals that we are able to grow in microgravity, we're able to utilize this technique called neutron crystallography and get a clearer picture. So with that, I just want to thank you very much. That is our report and experiences of growing protein crystals growth in space. Thank you.
Thank you, Adam, for that introduction and for everybody for having us here today. So without NASA And without the International Space Station National Lab and its managers at CASIS, low Earth orbit commercialization would completely stagnate. We're at what I think is an inflection point for continued development of commercial activity in space, and what CASIS is doing, what NASA is doing, is protecting that expansion and facilitating that expansion. And we at Made in Space have been lucky enough to be a part of that expansion of commercial activity in space. We originally started by developing a gravity-independent manufacturing technology, 3D printing technology that actually works in space. That was originally, kick-started via the SBI program and working with NASA. And now we have recently deployed this device that you see pictured here. This is the additive manufacturing facility. It is the very first commercial manufacturing facility off the face of the planet. For the first time, anybody in this room, government, private, individual, corporate, can actually utilize this hardware to send things to space, to do manufacturing in space, to further science, to more rapidly iterate and test in the microgravity environment, in a persistent microgravity environment. And this for us has been a really amazing beginning of what we have set out to do. And we've had incredible partners to grow this, and to be able to utilize the International Space Station as a proving ground, both for science and research and development, and also emerging business models. The AMF has been, operates under a user agreement with CASIS, and so we can explore and develop that business model by working with them. And we really think that that activity is driving the LEO economy, but there's a saddle point here. The way that we have made money off of space commercially since time immemorial as humans has been sending signals back and forth, sending ones and zeros back and forth. We've also sent a few people to space, a few spaceflight participants, but we at Made in Space believe that manufacturing in space is the real game changer for driving activity into space and driving and utilizing that new environment, and for those of us that are really big sci-fi geeks, actually making the promise of Robert Heinlein and Gene Roddenberry's dreams a reality. So that's half the reason we do this. So what I'm happy to announce today is a new program that we've developed to actually manufacture for the very first time high value to mass goods in space that have real markets here on Earth. We're developing a payload in cooperation with CASIS and with a private company called Thor Labs that will actually take high value heavy metal optical fiber and manufacture that in space. Why are we doing this? Well, there's a lot of research that indicates, going back about 20 years, that if you make this particular fiber in space, there are a lot of the impurities and a lot of the scattering that we have on the ground when you make this material is actually suppressed. So, and this material, it's called Z-bland, has a very wide transmission window and theoretically has a much better signal loss attenuation than sort of your traditional optical fiber, your silica fiber. So by making this fiber in space, we can suppress a lot of the things that cause it to lose signal, and create a very high value material in space. So we'll actually prepare the device here on the ground, load it up with the preforms, put it on a rocket, send it to station. There, it's installed, it runs for a little bit, manufactures the fiber, and then it's downmasked back here to Earth. It's studied and utilized, so we actually are making things on orbit and utilizing them here on Earth. And we hope that this is the first of many experiments like this and business models that are ultimately based in low Earth orbit and one day beyond, but providing value back here to people on Earth. Thank you.
Okay. Thank you all fascinating projects and obviously quite diverse. So well, I guess one thing we can talk about, what they all have in common, is I'm interested in the logistics of all of this. So when you're working on the platform of the ISS, obviously you don't have direct control of what's happening. So I'm wondering if you can describe a little bit what the workflow is like and what that requires of you in setting up your work to be either automated or to be able to control remotely, and what you require from the team on the ISS to support your work. So I don't know, maybe Andrew, you could tackle that first.
Sure. So the optical fiber payload that I was just talking about is our third payload that we have built and will ultimately send to station. And we've learned with each one of those first two payloads that we've put up about how to build it, how to make it very easy on the crew, because the crew has very limited time, and how to remotely operate our devices in order to make things really easy for NASA and really easier for the crew to operate those things. So the optical fiber payload, we're building that device now. We're going through qualification, getting our ConOps down with NASA, with a target to actually deploy that device to station in early 2017, and really it'll be just a box that goes up. The astronauts will unpack it, slide it into a rack, give it power, give it data, and then we'll actually remotely command it from the ground in a pretty autonomous fashion, pretty automatic fashion. And then once we're done, we'll let everybody know we're done, and they'll unplug the power and take the box out and put it back in the Dragon capsule, and it will come back to Earth. So just to make it as easy as possible on the crew, so that they can move on to other amazing science that's going on.
Joe, how much hands-on intervention do you require from the team on the ISS?
Yeah, I think in our case, our hardware is entirely passive, so it requires no astronaut time. Most of the work, or if not all the work, was done in the preparation part. So the preparation part is really trying to transport our samples to the launch site and to do the preparations on site. I think we had an amazing support team from CASIS to allow us to do this with the lab facilities and with all the logistics. I think the challenge was to meet with all the scrub schedules. A lot of the proteins don't last so long, so I think a lot of the experiments had to be designed that it would account for not one, but perhaps even two scrubs. And so I think most of our challenges was to spend time to try to estimate time of activations, and in the worst case scenarios, if there were several scrubs, will we still be able to launch our experiments? And if it goes longer, then we would have to redo it. But in terms of the flight, once it gets off the ground, it's entirely passive, and I think on our part, most of the work is done, is the Earth preparation part.
Interesting. Dan, when we spoke earlier, you were telling me some of the things that you've been learning about the capabilities of the crew on the space station, that in fact the astronauts are more than just glorified pilots, that they do have some serious lab skills. And I'm just wondering if you could talk about that a little bit.
Yeah, so certainly I had the opportunity to meet up and talk to Mike Barrett, one of the, I think, astronauts that have flown to the space station twice, actually himself a physician and internal medicine specialist. And he said as part of his training, he actually went through some lab skills. So he became essentially a glorified lab technician, being able to do manipulations in terms of pipetting, cell culture, and so on. In terms of the tissue chip technology, certainly the technology as we are developing it for use here on the ground is as much very dependent on microfluidic technology, and happy to hear that one of the CASIS implementers are actually specializing on microfluidics in space. So certainly we're going to be using that, and then fluorescent microscopy as well. One of the things that we are innovating in this space is the, as much as possible, is online ability to non-invasively sample both the fluids and the tissues embedded in this chips. So my anticipation is that there will be hopefully less engagement of the astronauts, or in terms of hopefully being just a turnkey, pushing a button and then letting the fluidic pump and the cells have their way, and then occasionally having sampling going on.
So all of you, I believe, require both payloads to go up to the ISS, and then you need materials returned to you. So I'm just wondering what your experience has been with scheduling and coordinating launches, working with CASIS. I know the Flight Opportunity Program has been important for that. And is the schedule, is the flight schedule conducive to your research and to your future commercial efforts?
Yeah, I mean, I would say that you know, the short answer is yeah, the schedule works with our business model. But really, it's our job as a commercial space company to find business models that work with the current operations. You know, we have this really, I think there's this really amazing kind of development path that we've been lucky enough to take, and it starts out on, you know, parabolic flight aircraft, and then we prove that up, and then we can, you know, and then we take that to ISS, and the goal is to develop one or more business lines that eventually can survive and thrive on commercial platforms in the future, or on free flyers. So we you know, we sort of come and say, hey, like, let's figure out how to make something work with ISS, not try to say, hey, ISS, you know, do X, Y, and Z for us.
Joe, do you have any? What's your experience been with that?
I agree with him, and you know, I think the secret is to be as flexible as possible. If you understand that flexibility is a criteria, then your scheduling can work with you. And as I said, the only challenge we had was in cases of scrubs, for whatever reason, weather or, you know, mechanical reasons. I think the experiments have to be designed where you would have to expect these things. And the flexibility, the plasticity is very, very important in your experiments. If your experiment is designed to be rigid and has to be done, you know, in a very, very with narrow restrictions, it's not an experiment to do. So, but I think the schedule does work very well. As I said, you know, with CASIS, with supplying the labs, and, you know, with all their support, I think the user, with its degree of flexibility, can work very, very well with a very consistent schedule for commercialization and academic research.
And Dan, you're still some time off from launch. You're actually in the process of adapting our terrestrial organs on chips technology to for use in microgravity, and working with researchers who are doing that.
That is correct. But having said that, the main focus right now, as part of our partnership with CASIS, is actually to have sort of like a late 2018 launch date. And so part of that is part of the 2-year of the program is to adapt the technology that we have for use here on Earth for use at the space station. And I think, and part of that, I think, is actually even prior to making the announcement in public, which was announced Monday, CASIS and NIH have already had a webinar back in April to hook up our tissue chip developers with CASIS implementers, just to make sure that the technologies and for use in space are actually being deployed for, you know, using the tissue chip technology. So we have, you know, sort of like a 2-year launch date, but we're thinking ahead and planning ahead, and hoping that
that flight and then the subsequent flight 2 years later. So it'll be 2 flight dates that we have arranged through CASIS for access to the space station.
So Joe, as I mentioned earlier, is this on? Okay. You've been flying research, well, not personally flying, but sending up research into microgravity for quite some time now. I'm wondering how you've seen the actual hardware evolve in that time, and how much sharing there is amongst researchers in that field in iterating those designs and sharing those developments with others.
That's an easy question, because when we started with the space shuttle, in terms of getting statistically relevant results, it was much more challenging then, because the hardwares were built where you could only fly maybe 2 or 3 to a dozen samples at most. And so if there was any type of anomaly, you would kill a whole experimental data set, and your conclusions would be really skewed to thinking from success to failure. Today, I think the hardwares that are available to us allows us not to have only 10 or a dozen, but hundreds of samples. And that allows us to look at statistically significant results. And we are able to make very more concrete conclusions out of those. So hardware's looking at 96 well setup. In our case, we put up 400 samples. And out of 400 samples, 75% were successful experimental results. So what I don't talk about is that other 25%. But realistically, those 25% were anomalies from preparations and things like that. But if we would have flown 5 or 10 samples, you wouldn't get this case. In terms of outreaching to collaborations and things like that, because we have an excess of sample space, we're able to work with collaborators and include more diversified conditions, which increases our chances of success. And so yes, today is much more easier to fly meaningful experiments where results can be much more representable to what microgravity can influence.
Interesting. And I should mention the Nature Publishing Group has just announced that they're launching a microgravity journal as well, which I imagine will be a useful forum for sharing developments in that research. So we are talking about business here and commercialization. So you obviously all have different lenses through which you evaluate ROI on this kind of work. So Dan, obviously you told me you're answerable to the taxpayers ultimately and have to make the case that this is a good investment of taxpayer money. Joe, you're doing a combination of academic and commercial work. And Andrew, you're obviously trying to make a high value product that will justify your work even without subsidies and launch subsidies from NASA. So I'm wondering if you could just offer some thoughts about how you think about ROI and how you think about the time frame for a return on your investment here.
Yeah, we really look at try to identify products and services that can survive in at a price per kilogram to launch and return area that are going to be economical. So that means you have to look at the cost of development of the hardware to do the manufacturing. You got to look at what do you need on orbit to do it. What are the costs of your cost of goods sold, raw material. You got to look at the cost of launch, and then and launch is a word that we use a lot in space, but it's not just launch. It's preparation, integration, getting the thing off the Earth, and then getting to an environment that is hospitable, and then returning to Earth. Because today, all the economic activity that we are interested in and that we think is viable is tied to Earth. One day, I am super excited to be manufacturing with platinum from asteroids that and make things for people who were born in Leo. That would be amazing, but we're not doing that today.
So so that you have to close the business case on, and we're space people, so we think in dollars per kilogram to close the business case on that. So that, because we ultimately want to, to progress beyond the national lab to commercial stations.
Joe, can you talk about your perspective on that? So how do you evaluate your return on this kind of work?
So this kind of overlaps to your previous question. And what I didn't mention, besides the hardware capabilities, is also the duration of time in microgravity. Before, we only could spend maybe up to 2 weeks on a shuttle, where now we could spend, 6 months to a year. So with that time of duration, we could do more meaningful experiments. And so with commercialization, with investments, at least the clients that we work with, investors or customers now can actually see more effects of the experiments that we've gone up. And even though the duration is much longer, and so you could see more effects of the microgravity, and so there's a higher chance, and there is a higher chance that you get a product out, you get a result out. So in that case, I think that at least the customers we work with, they're much more enthusiastic that their products or their experiments will be going up for a more lengthier time.
Dan, and so you need to present a case to Congress that this is something that we should be doing. Just in a nutshell, what kind of time frame are you talking about in seeing results, and what kind of results are you?
Yeah, so essentially, the short answer is when Congress authorized the formation of NCATS, it was essentially to address the issues on drug development that is currently plaguing the drug development. Essentially, the drug development pipeline has dried up. And so that typical drug development takes anywhere from, and Joe's probably familiar with this, anywhere from 10 to 15 years. And so part of the Cures Acceleration Network that I lead at the NIH is to cut that at least by half. And so our schedule, or our goal, is to, in fact, my director always says, if there's nothing tangible within the next 3 to 5 years, then your project should not be funded by NCATS. And so our goal is along those lines. So our ultimate return on investment is ultimately the betterment of human health in terms of better therapies and better diagnostics, hopefully with a faster turnaround time.
Right. And I think it's just important to mention, Joe, you and I talked about this a little bit, but success is defined differently from the perspective of the public versus a perspective of a scientist who's doing sort of basic research. So, your average person may think, oh, we don't have a new drug coming out of this program yet, but the drug development pipeline is actually very, very long.
Look, there are 3 big government support projects that I could think of which came out with a lot of new technology and innovation. The Genome Project, which is heavily funded by NIH, the Protein Structure Initiative, which is to determine as many 3-dimensional structures of protein, also funded by NIH, and the Protein Crystal Growth, funded by NASA previously. All these 3, what they have in common was that the proposal had suggested that by doing this, you have a cure to cancer, a cure of these diseases, and this appearance of overpromising, and that's how the public perceived it. But in reality, science is very incremental, and it's incremental in a way that you invent, you create new technology, and from that, more technology is given. So from the Genome Project, I would say, no, we don't have a cure to cancer yet, but what we do know is a great and enormous understanding of gene regulation, how our body for diseases that are associated with certain pathways. In terms of Protein Structure Initiative, we have an immense understanding now about the evolution of structures and about what's important for drug designs and what are the important sites of these. No, we don't have a cure to Parkinson, Alzheimer, and protein crystal growth, what NASA had funded. I think it was a great investment because out of that, you have new technologies in detectors, hardwares, and also basic cyclotron lines preparations that would not have been possible otherwise. And I think with all this, I think these investments are worthy, and you know, the public, I think, needs to be reminded that there's a lot of inventions, innovations that have come out in pursuit of the final goal, which is to ultimately find a cure to one of these high-impact diseases.
So I do want to leave time for questions, but I also, while I have you three very smart guys up here, just wanted to do a little bit of brainstorming. So we know that some of the most impactful innovation really happens at the intersection of disciplines. So, you know, organs on chips come out of semiconductor manufacturing technology combined with stem cell technology. Synthetic biology came out of the merging of molecular biology with computer science. So you're going to have this very diverse base of users and researchers on the ISS platform. I'm wondering just what your thoughts are about encouraging synergies between these different kinds of technologies. So obviously 3D printing is used for materials ranging from plastic to cells. And we were actually talking the other day about, you know, potentially printing some food for the astronauts to get them something a little more interesting than what they're used to. So just kind of taking it out there a little bit, just wondering what you think about using some of the resources that are available on the ISS to do maybe some wildly different things that bring together some of these different disciplines and tools. Who wants to take that?
I guess I'll start. Yeah, I mean, I think that the you know, being able to manufacture things on orbit and you know, and augment or plus up the other experiments that are going on station is a natural synergy. And then if you extend that a little bit further, being able to make do bioprinting, maybe make chips on tissues on chip in space and see what that looks like, or make or a lot of the other kind of very interesting bioprinting and vascularized tissue challenges that are out there today, to me is a really exciting prospect.
Joe?
I have nothing to add.
So actually, I was in a meeting a couple of weeks ago with members of the National Academy of Sciences who just published a report on convergence, taking multidisciplinary approach. Essentially, it's a team science approach, taking engineering, physics, mathematics, computational biology, and life sciences in terms of addressing, and capitalizing on the developments in these fields. And tissue chip technology was actually one of the examples that the authors of this report have put together. I think we will continue using a multidisciplinary approach, a convergent science approach on developing tissue chip technology, and I think that's the opportunities at the International Space Station is going to be ripe. I think Dr. Topol even mentioned space genomics, being able to, because one of the advantages that we have with this tissue chip technologies is it's heavily reliant on stem cells. And so individuals, cell lines can actually be put in terms of actually having a precision medicine approach. So we can potentially identify individuals who are, have a better aptitude for long duration in space. You can actually identify those who are more susceptible to damage in their DNA from prolonged exposure to radiation. One of the things that I am intrigued by, and mentioned it to Andrew is that, probably one of the things that bioprinting can be used for, having heard one of the astronauts say that, after 3 weeks, food becomes bland. And so, potentially you can bioprint a steak or a hamburger using muscle cells derived from cows or lamb or whatever lamb chops you want. So it not only does it improve the quality of life of the astronauts, but obviously will be a viable option for eventual colonization of people in space.
Right. Right. So if people do have questions, there's a microphone set up over there. Great.
Yeah, being constant challenged, I guess it's for Dr. Joe. Question is if it would be a benefit or if there's any plans or thought about setting up a crystallography unit to the space station. I understand there are some organic compounds that are weakly held together by hydrogen bonds that might actually survive the return to Earth, and would that help a lot in terms of both accelerating the speed, and is it practical to think about doing something like that?
It's a good question. About 15 years ago, that was proposed to NASA. They were actually on the way to build a diffractometer up in the space station, and that got canceled. I would say that back 15 or 20 years ago, that may be very, very effective. I think now, because of the developed cyclotron line, the fine beams, the sensitive detectors that are built on the ground, I don't think that is as needy anymore. And that all came because of what I previously said from all these other projects that has developed along the way. And that's for X-ray. So let me distinguish X-ray, as I said before, that only tells you partially of all the atoms, and that's most of the structures. Now, in terms of neutron structures, that is a different story. Now, it's unfeasible to have a neutron source up in a space station because that needs to be coupled to a nuclear reactor. But certainly, if there is a way to at least analyze or look at the crystal growth property up in the space station before it comes down, I think that would be a great help. But in terms of what you're asking, would it be productive to have a diffractometer up there? I think there's an argument to have one. The cost is of concern, whether the amount of information that you get out of it would be worth the cost invested into it.
Good morning, Rich Jackson, Lockheed Martin, and really want to thank the panel for the discussion here. I wanted to follow up on something, Adam, you'd asked about logistics. In commercialization, speed is of the essence. And, we're talking about a lot of things that a lot of folks have talked about from genomics on through and the studies that you're involved in. Question I have for the panel is the speed and adjustment of the data you're getting from each sample you fly to station. Is the flight rate correct for you, or is there something the ISS team can do to give you more flight opportunities to speed up the research that you're actually focusing on right now to speed up where we get to eventually?
I think for us it's just right because we're just getting started. I'm sure there will be a demand subsequently and after 4 years of the program once it's been running.
I'd say that's another one of the things that, like I said earlier, that I feel it's incumbent on us as a commercial company to adapt to the playing field that exists rather than asking the playing field to change for us. With the optical fiber in particular, that's a field that it is not uncommon for people to place an order and wait 3-6 months, a year to get their material. So that fits very well. And then as we progress that program, we hope to build hardware that we will fly and refly
at a rate that will service what we believe the existing market is and what potential markets are at the flight rate that exists now. Certainly more ships going up and down will be great, but that's not something that we need to survive and thrive.
Okay, and really the focus was on the research. If you had flight rate or flight opportunities already queued up that you could adjust quickly and then slide right back into it just like you would do in a lab, would that speed things up? And it sounds like you guys are saying no, the rate you're doing, and when you get it back down, the time to reschedule a flight is working for you at the current time. Thank you.
Hi, I'm Jeff Fitzsimmons, retired engineer. This is a question mainly for Joe and Andrew. Back in the 80s and 90s, before the space station and while the shuttle was flying, NASA developed these entities called commercial space centers. It's kind of a precursor to CASIS in an attempt to commercialize and develop products to be made in space and brought back to Earth or made in space and looked at in space. And I'm just wondering if you looked at those commercial space centers, some of which still exist today, a lot of which have already folded and did not succeed. And I was wondering if you looked at the results and the outcomes and the lessons learned of those commercial space centers, and particularly the Z-band fibers. I think they looked at those for a long time, and I know that they did a lot of studies with the protein crystallization. Did you look at the outcomes of those, and do we need something like that perhaps to foster commercialization today?
Yeah. So I personally have not looked at it recently. I do know about those commercialization centers. In terms of protein crystal growth, as I alluded earlier on, the challenges was really trying to get a statistically significant results where you could fly many, many different samples, and that was not possible back then, where you only flying a few parts. So in terms of the opportunity to commercialize anything around protein crystal growth was very, very limiting. And I think even up to now, these centers have put PCG really in the bottom of the list, if you will, in terms of having any types of useful commercialization until we start getting more results back as of now, where we're able to fly more experiments.
For us, the activities that those centers carried out is something that we've looked at significantly and looked at pretty in depth, rather. And some of that work and work done in other portions of NASA and the Air Force was the motivation. It was the thing that allowed us to get comfortable with putting our own money and time and effort into trying to make this optical fiber manufacturing facility in space. So that has been invaluable to us, and we've even had some aspects of that work kind of been pointed out to us by people that used to work at those centers, or civil servants that work with us now, like Nikki Werkheiser at Marshall Space Flight Center has pointed that out to us a lot, and we really value that direction. Going forward, I think that we are at this commercial tipping point or inflection point, or however you want to phrase it, where there are a lot of companies like Made in Space and NanoRacks and Space Pharma and these other entities that are kind of standing up and saying, hey, we can take these maybe low TRL or middle TRL technologies and invest in them and turn them into business models and sustainable business models. So I think that if organizations are continuing to kind of prime the pump with doing that sort of work, I think that's completely appropriate and awesome and should be encouraged. But today maybe there's a little bit more nuance in trying to figure out how much pump priming there needs to be before to encourage commercial innovation and activity in space, without unintentionally smothering companies that are saying, okay, that's far enough along for me to put money into that and me to go do that. Because ultimately, I think that's what we want. We want to get it to a point where commercial interests can pick it up and run with it, And then from there, it sort of fractures out, and other people are inspired by that, and additional business lines are developed by those entities. And I hope that's the economy that, and the ecosystem that we're trying to create.
Thanks, Andrew. We are out of time now, so if you have additional questions, please come and find these guys after the panel. I want to thank all three of you for a fascinating discussion today, and thank NASA and CASIS. And all the CASIS people are wearing these cool black polo shirts today, so if you have additional questions, find the people in the black shirts. Thanks again.